So we're going to be looking at DNA replication in prokaryotes. So in DNA replication, in this picture, we have bidirectional replication happening, meaning that's going in both directions. So we have two origins of replication, which is a sequence on the DNA where replication will begin. As we can see, if you look very closely at the orange lines, which represent our new replicating DNA, we have one continuous strand of replication that's going into the fork, and then we have discontinuous replication that's heading away from the fork. So the discontinuous strand of replicating DNA forms what we call Okazaki fragments, which means that there's basically several different fragments of replicated DNA that we later connect. So let's take a look at all the proteins and enzymes involved in DNA replication. We have helicase, which is shown by the brown comma, which is actively unwinding the DNA. Next, we have gyrase, which is shown by the green donut shape, which is keeping the DNA from knotting up by relieving torsional stress. And basically, as the replication bubble widens, DNA has a tendency to knot up, and gyrase is just preventing that from happening. Next, we have primase, which is setting down RNA primers for DNA to replicate off of, because DNA polymerase 3 needs something to add on nucleotides to. It needs a 3' OH to add on to, which we'll talk about more later. Next, we have DNA polymerase 3, which is just basically the protein or the enzyme that is actively synthesizing DNA. It's adding nucleotides to the last 3 OH of the primer. So we're going to take a closer look at what's happening at an Okazaki fragment. So we have primers, which are shown by the small light orange boxes that were put down by the primase earlier, which is what DNA polymerase 3 adds onto. And since these are the Okazaki fragments, we have multiple pieces that we have to attach. So DNA polymerase 1 comes in and removes the primer, and then ligase comes in and attaches those two Okazaki fragments, making one continuous strand. Then we have these little bundles of pink dots, which are representing the single-stranded binding proteins, which keeps DNA in a single-strand state so that we continue to replicate the DNA. So we're taking an even closer look at our replication here. We have old and new DNA shown by blue and orange. As we're adding on nucleotides to the 3' OH of the last one, we are cutting off 2 extra phosphates from that new nucleotide, as you can see at the bottom here. And this is why we need the primer whenever we're replicating DNA, is because DNA polymerase 3 needs something to attach that phosphate to. So it needs the last 3 OH of the nucleotide there to add onto. These nucleotides are held together by phosphodiester bonds, as shown on the old strand of DNA. So we're asked what would happen if DNA polymerase 1 could not perform its 5' to 3' exonuclease activity or function. So that function is to remove primers. So basically, DNA polymerase 1 would not be able to remove these primers, which means that ligase could not come in and connect these Okazaki fragments.